Hello everyone, here I'll introduce the paper submitted to iMeta. The title is Pan-Cancer Analyses Reveal Molecular and Clinical Characteristics of Cuproptosis Regulators. The cytotoxic mechanisms of metal ions, including copper, are suggested to mainly rely on oxidative stress. However, a recent study published in Science challenges this conventional view. That study revealed that intracellular copper accumulation can induce a novel regulatory cell death mechanism via the aggregation of lipoylated mitochondrial enzymes and the destabilization of the iron-sulfur cluster proteins, which is termed as cuproptosis. To identify the genes involved in cuproptosis, this study identified 10 genes that can regulate cuproptosis, including 7 positive regulators and 3 negative regulators. To provide an initial overview and reference point for future studies, we used big data to comprehensively assess the molecular and clinical characteristics of cuproptosis regulators in 33 tumors from a multi-omics perspective. Firstly, we analyzed SNV and CNV data from pan-cancer samples and portrayed SNV and CNV landscapes. Overall, there is almost no changes in SNV. The CNV frequencies are very low for almost all cuproptosis regulators, except CDKN2A that showed CNV frequency in up to 14% of the tumors. We also characterized the somatic alterations of these cuproptosis regulators in each of tumors to better understand the landscape of different tumor types. Overall, the mutation and amplification frequencies are low in tumors, but a high percentage of CDKN2A deep deletion is observed in many tumor types. Among them, more than half of the GBM samples showed deep deletion of CDKN2A. Differential expression analysis of paired normal and tumor tissues revealed that cuproptosis regulators were aberrantly expressed in 17 tumors. We performed unsupervised consensus clustering based on regulator expression for all samples. All tumors were distinctly divided into 2 different sample clusters. Compared to Cluster 2, Cluster 1 had lower expression levels of CDKN2A but lower expression levels of others. We also checked the distribution of all cancer types in Image C. The expression of any individual regulator can hardly reflect the overall level of cuproptosis. Therefore, we first proposed the cuproptosis positive score, cuproptosis negative score, and cuproptosis activity score based on the expression levels of cuproptosis regulators. We found that Cluster 1 had significantly higher cuproptosis positive and activity scores. Survival analysis showed that Cluster 1 had a significantly better overall prognosis than Cluster 2. Methylation in the promoter regions of genes largely regulates the gene expression. As shown in Image A, the methylation levels of regulators varied in all 16 tumors and methylation alterations in regulators were heterogeneous in different tumor types. Furthermore, we analyzed the correlation between regulatory methylation and mRNA expression in 33 tumor types. In most tumor types, the promoter methylation levels of FDX1, DLAT, GLS, and CDKN2A were negatively correlated with the mRNA expression levels. Survival analysis revealed that the methylation levels of cuproptosis regulators were correlated with the OS in 17 tumor types and that the correlations were tumor type dependent. miRNA, IncRNA and transcription factors are also important regulators of gene expression. In this study, we have fully explored miRNAs, IncRNAs and transcription factors, which may regulate cuproptosis regulators. The results can be found in supplementary materials. In addition, we found some miRNAs, IncRNAs, and transcription factors targeted multiple regulators. So we constructed the 3 regulatory networks. Cuproptosis regulators and scores have different prognosis values in different tumor types. For example, higher cuproptosis activity is related to poor OS in HNSC, and to better OS in UCEC. In addition to OS, we also tested the relationship between cuproptosis and DFI and PFI. The specific tumor-related pathways involved in cuproptosis remain unknown. Therefore, we first inferred the enrichment level of 50 cancer hallmark gene sets in all tumor samples. Cuproptosis activity was positively correlated with half of the hallmark gene sets in most tumor types and negatively correlated with the other half. Notably, cuproptosis activity was significantly positively correlated with oxidative phosphorylation and fatty acid metabolism in all 33 tumor types. Strikingly, cuproptosis was negatively associated with immune-related pathways, including the inflammatory response. To better reveal the intrinsic link between cuproptosis and tumor immunity, we first inferred the overall immune and stromal infiltration levels in all tumor samples. Cuproptosis was significantly negatively correlated with the overall immune and stromal cell infiltration levels in most tumor type. We calculated the correlation between cuproptosis and the levels of immune cell infiltration. There was a heterogeneous correlation pattern between cuproptosis and tumor infiltrating immune cells. Cuproptosis was negatively correlated with most immunomodulators in most tumor types, which is consistent with previous results. A previous study classified all samples from 33 tumor types into 6 immune subtypes with different immune characteristics in a pan-cancer context and were widely recognized. As shown in image E, C4 tumors had the highest level of cuproptosis activity, whereas C6 tumors had the lowest cuproptosis activity. After establishing the association of cuproptosis with numerous cancer hallmark pathways and immune-related characteristics, we aim to understand whether cuproptosis could influence patient response to chemotherapy, targeted therapies, and immunotherapy. We observed significant negative correlations between cuproptosis and the IC50 of 39 drugs but failed to observe a positive correlation between cuproptosis and the IC50 of any drug. We collected 2 immune checkpoint inhibitor cohorts to investigate the correlation between cuproptosis and immunotherapy outcome. Cuproptosis was found to be negatively correlated with the tumor immune score, stromal score, estimate score and PD-L1 expression in IM vigor cohort. Importantly, in the 2 cohort, patients with high cuproptosis activity had a better OS after immune checkpoint treatment than those with low cuproptosis activity. Here is the summary of this study. You can find more information and results in the supplementary materials. Thanks.